Thank you so much, Rochelle, for the introduction. And thank you all for coming, and thank you to the organizers for allowing me to speak. Today, I will be following up some of the lovely talks we had this morning, talking about some of the things that we do at Cayman Chemical and what we've learned about planning different types of immunopeptidome profiling experiments and how to ensure that you get the best possible information from these experiments. We've had some really excellent background from Dr. Leung this morning and other speakers, but just for a perspective, over the past 50 years of cancer research, we've come a long way, but there is still really far to go, as we saw this morning. The majority of the research has focused on finding better toxic chemicals to selectively target cancer cells. But the advent of immunotherapy has turned cancer treatment on its head and led to some of the most impressive advances we've ever seen. The immune system is made up of highly specialized cells with exquisite specificity, along with a memory of previously encountered antigens. Current immunotherapies seek to help the immune system learn to recognize and eliminate cancer cells, which has begun to reap the rewards of safer and more effective and durable treatments. An important principle that makes immunotherapy to cancer possible is the intrinsically disordered protein expression of cancer cells. These disordered protein signatures can help the immune system differentiate cancer cells from normal cells. Every cell, normal, tumor, infected, dying, presents peptides derived from endogenous proteins in MHC class I molecules, depicted here by the blue protein with the red peptide. These peptides are typically 9-mers and are how T cells and the TCR, here in green, determine if that cell is normal. Is it self? Is it virally infected? Is it expressing unrecognizable proteins? If the T cell thinks anything is wrong, that cell is targeted for elimination. Since cancer cells frequently have intrinsic genomic instability and accumulated mutations, there is a possibility of those mutated proteins being broken down for presentation in MHC, and a T cell recognizing them and initiating immune-mediated destruction of the tumor. If we can exploit this to develop treatments, we can induce the body to kill its own cancer. So how does immunopeptidome profiling fit in? Immunopeptidome profiling aims to sequence the peptides found in MHC complexes. This allows us to directly determine what peptides have the potential to stimulate immune responses, as presentation in MHC molecules is requisite for T cell responses. The vast majority of peptides identified in immunopeptidome experiments are derived from normally expressed proteins. But if neoantigen identification is the goal of your experiment, we are looking for a needle in a haystack. The term neoantigen encompasses two different kinds of peptides. Tumor-associated antigens are a part of the normal human repertoire, but may not be normally expressed in the tissue the tumor is derived from. Identification of these would require comparison of tumor tissue with normal tissue peptide lists. Tumor-specific antigens, on the other hand, are derived from completely new proteins, such as indels, translocations, other mutations that we heard a lot about yesterday and earlier today, and thus may provide greater potential for immunogenicity. Immunopeptidome profiling can be applied to cancer research in many different ways. Personalized and universal cancer vaccine development that we heard about from Michal yesterday relies on the identification of neoantigen epitopes specific to either an individual's tumor or common to one or more tumor types. Similarly, adoptive cell therapy targeting neoantigens requires tumor-presented targets. Immunopeptidome profiling can also be used to evaluate the efficacy of therapy designed to be used in combination with checkpoint inhibitors, as we heard earlier from Greywolf. Treatments such as chemoradiation or oncolytic viruses, or even ERAP1 inhibition, may increase neoantigen presentation and thus sensitivity to checkpoint inhibition. The ability to predict neoantigens from much less expensive and time-consuming techniques, such as nucleic acid sequencing, is a valuable prospect, but these prediction algorithms are only as good as the training data sets used to teach them. Again, we heard a lot about that this morning. The workflow for immunopeptidome profiling is pretty simple overall. First, we lyse the cells or homogenize the tumor sample. Then we immunoaffinity enrich the MHC complexes using antibody conjugated resins. Peptides are gently eluted from the bound complexes and subjected to LC-MS/MS by our talented colleagues at MS Bioworks. From this, a list of peptides is developed using relevant databases. I will discuss some key considerations for each step in this process. When making our way through the immunopeptidome profiling workflow, the first stop along the way is input. One potential input is tissue, whether it's tumor or normal, whether mouse or human, or biopsy or xenograft. The big question is always, how much material do we need? The best answer we have is usually more is always better. Ideally, we get SNAP frozen tissue samples of at least 100 milligrams. Tumors can vary dramatically with respect to the recovery of peptides, as shown in the graph on the left. Here, a cohort of tumor samples were homogenized, and class 1 and HLA-DR expression was measured in the homogenate by ELISA. The respective IPs were performed, and the number of peptides recovered from the sample is shown versus MHC concentration. The samples with the most MHC do not always provide the most peptides. There are a lot of possible reasons for this, such as sample handling, the cell types in the tissue sample, the clonality of the cells, or many other variables. Similar variability exists among cell lines. We have tested various mouse and human lines and usually recommend starting with at least 100 million cells. In some cases, this won't be enough to get a complete picture of the immunopeptidome, or in some cases, many fewer cells may be required. The graph to the left is the starting concentration of MHC class 1 in various human cell inputs tested by ELISA. With the exception of the DCs, all of these were lysed at the same cell density in the same buffer, showing the extreme variability in potential starting material. For some model cell lines, such as the breast cancer shown here, 100 million cells was not enough to see the desired epitopes, while in other models, as few as 5 million dendritic cells may be sufficient. Another consideration here may be the expected frequency of the sought-after epitopes. Rarer epitopes will require higher input. An important thing to note here is that immunogenicity cannot be used as a surrogate for frequency, as many other factors contribute to T-cell recognition and response beyond epitope frequency. Tissues and cell lines are not the only potential inputs for immunopeptidome analysis. We did a small study looking at soluble MHC in small size plasma samples from cancer patients. We found by ELISA, shown here on the left, that there was a significant amount of soluble MHC class 1 circulating in each of these subjects, and peptides were recovered from all of them. This could potentially be an interesting approach to query tolerance to neoantigens. A critical choice for experimental design is also the choice of antibodies for the immunoprecipitation step. For IP from human samples, the most commonly used antibody is W632 because it has broad specificity across human class 1 molecules. HLA-DR can be efficiently captured by the antibody clone L243, and the clone IVA12 conjugated resin can pull down other class 2 molecules. Other antibodies to specific classes of HLA molecules exist, but have not yet been optimized in our system. It is important to note that in many cases, these IPs can be performed serially to capture both class 1 and class 2, or multiple different alleles from the same sample. The mouse antibody situation is substantially less straightforward, and shown here is a typical workflow. M142, the first antibody shown on the left, is the closest thing we have to a pan class 1 antibody. And in our experiments, it is able to pull all class 1 molecules from BALB/c mice, as shown with the logo plots on the bottom. But it only pulls down significant amounts of H2KB from BLAC6 mouse samples, which we discovered by performing Gibbs clustering of results from BLAC6 experiments and noting the distinct lack of an H2DB motif. Luckily, we have an antibody called 28148S that can immunoprecipitate H2DB from these samples when employed serially. Motif diagrams for each of these peptide sets are shown here. Frequently, MHC class 2 immunopeptidome analysis is desired from the same samples, and this can also be performed serially with the class 1 IPs. The key takeaway here is that many things are possible, and so it is very important to have a very detailed discussion of the goals of the experiment so that we can help you design the study for maximal output. Another thing to consider is how any treatments will impact the peptide number. For instance, interferons are sometimes used to upregulate MHC expression, but the question remains how the stimulus may change the repertoire in unanticipated ways. Signaling changes have the distinct potential to be reflected in the immunopeptidome. That being said, here we're showing HCT116 cells that were left untreated on the left or treated with interferon gamma on the right. From each of the peptide lists, gene ontology analysis was performed to assess any global shifts in protein repertoire. And in this case, none were actually seen. However, this is only one cell line and others may respond differently. Conversely, sometimes the goal is to find a treatment that will alter the immunopeptidome and potentially increase the likelihood of generating neoantigens. Radiation treatment, chemotherapeutics, and oncolytic viruses may be employed for this purpose as much as to directly kill cancer cells. It is possible that these treatments will affect MHC expression levels, and any resulting increase in peptide numbers may be due either to the increase in MHC at the surface or to an upstream mechanism affecting the repertoire. This may not be a problem, but it is important to note that the number of peptides identified cannot be normalized to the amount of MHC. For example, we performed a study investigating whether commonly used chemotherapeutic drugs could affect the MHC immunopeptidome and possibly sensitize cancer cells to immune-mediated killing as part of the mechanism of action. When we tested the MHC levels of the treated cells by ELISA, we found that all of the treatments increased the class 1 expression as compared to untreated cells. This information is interesting by itself, but also underscores that any potential increase in peptide number with treatments may be a result of greater MHC expression as opposed to generation of new antigens. Another critical consideration for experimental design is the expected output of the experiment. What mass spec sequencing of peptides essentially provides is a list of masses that must be matched to known proteins before they have any meaning. This means that we need to know what we're looking for in the immunopeptidome in order to find it. The default database against which data are searched is the canonical species database from UniProt, which will match all normal proteins. Beyond this, we can ask for the analysis program to search for single amino acid changes from the normal database to address neoantigens derived from point mutations. However, any more expansive mutations, such as indels, splice variants, translocations, cannot be found using these tools. This would require addition of mutated sequences from either transcriptome sequencing or exome sequencing, as we heard in great detail this morning. In the experiment with the chemotherapeutics, we only use the canonical human database. As expected and shown on the left, most peptides were common to all the conditions. Interestingly, the treatment groups shared more peptides with each other than they did with the untreated group. On the right, we also looked at single amino acid mutations from the canonical human database, which is as close as we could get to neoantigen identification without sequencing transcripts. The next step here would be to sequence transcripts to find larger mutations and compare any of these back to the peptide data. For any experiment like this, neoantigens that did pop out would need to be validated in either T-cell activation studies or further mass spec-based immunopeptidomics. For mass spec-based target validation, there are several possible methods to consider. For class 1 peptides, which are derived from endogenous proteins, the peptides of interest can be overexpressed in the context of the protein they are normally found in, or as a series of tandem mini-genes. Timing of expression may be critical in this case, as it is necessary to allow time for the construct to be expressed and the protein to be broken down by the proteasome, followed by peptide loading in the AR. In some cells, cross-presentation of antigens may be possible, meaning that cells take up exogenous antigens via pinocytosis and then traffic them to the ER for class 1 loading. Alternatively, soluble peptides may be incubated with cells for a short time to allow direct peptide exchange at the cell surface. Class 2 peptides are derived from phagocytosed proteins, and ensuring the efficient uptake of the target is a critical point. Soluble proteins are not taken up very efficiently, so coating latex beads with the target protein may be a more effective method of ensuring uptake and processing through the endolysosomal system. One potentially useful tool to approach the immunopeptidome analysis is using monoallele expressing cell lines. There are a few lines available which have little to no endogenous MHC expression, and which can be transfected with single alleles of MHC to create a cell line in which all peptides are specific to that MHC allele. If targets are then added using some of the strategies mentioned earlier, the ability of these peptides to bind to that specific allele are tested. In our experience, there can be dramatic variability among alleles expressed in the same cell line. The graph on the right shows a series of different monoallele samples generated at Cayman, looking at peptide numbers versus MHC expression level quantified by ELISA. It is clear that in the same cell type, there are certain alleles that can bind high numbers of peptides despite low expression, and others that cannot bind many peptides despite very high expression. We are currently investigating ways to optimize and boost recovery with some of these more challenging alleles. The introduction of a target adds another variable, and it would appear that timing, again, is key. Optimal timing of target availability to MHC in these monoallele lines may require stable expression of the target, along with transient expression of monoalleles in that stable line. There we go, sorry. Cayman immunopeptidome profiling services can provide a complete solution to complement your neoantigen research. From experimental design to antibody production, cell culture, transfection, and immunoprecipitation, as well as expert mass spec with our colleagues at MS Bioworks, we can meet your needs. The larger contract services organization can draw on hundreds of skilled scientists to address your research problem, but we are still small enough to be able to provide a personalized and flexible approach to each and every project. We believe that a key component to solid science is quality data and reporting, and strive to maintain superior standards every day. With that, thank you for listening, and I would be happy to take any questions.
Thank you, Julie, so much for a great presentation. So we'll go over now to the first question, which is, so what is the cost of the immunopeptidomics as a service?
Well, the cost of immunopeptidomics can vary quite dramatically, actually. We, as I mentioned, offer a really complete list of services, but In terms of the cost for immunoprecipitation and mass spec analysis, it essentially starts in the range of $3,800 per sample.
Perfect. And then on to the next question for you. What kind of samples can be analyzed for immunopeptidomics?
We can analyze all kinds of different samples. As I mentioned, we can do tissue samples from either human tumors or mouse tissue. We can use cell lines. We have a long list of cell lines that we can grow in-house, and cell lines which we can transfect in large scale. There are other kind of different sample types that we can analyze as well. We did a small study with the soluble MHC from plasma, and that was a really interesting different approach to looking at MHC.
Perfect. And we have another question for you. So how long does it take to get from sample to peptide lists?
If we are provided with a sample, it typically takes about 4 to 6 weeks to get from all the way from a snap frozen pellet to a list of peptides provided.